Good morning to everybody. It's a pleasure for me giving now like a warm welcome to this second day of this international symposium about microbiomes in different contexts and with different approaches. For this session that is starting now, we will talk about methods of collection, preservation, and research. We will have several wonderful presentation, I hope, and information from several speakers from around the world, from Chile, Switzerland, Germany, the US. So I should remember you that we have between 10 and 15 minutes. That means that a maximum of 50 minutes, that if you will not have any time for question and will have to keep them for the end of the session. Our first speaker now is Dr. Eduardo Castro from Universidad de Talca in Chile. He will give us the talk Composition, Taxonomy, and Functional Diversity of the Microbiota in Asthma in Chile, US, and Portugal. And for me, it's a very, very nice initiative to have the possibility to introduce you, Dr. Castro. Please.
Thank you, Nicole. Let's see if this works.
Perfect. But not that, oh, that's okay.
Perfect. Okay, so I guess I'm some sort of a leftover from the microbiome session yesterday, because I'm not going to talk about methods necessarily. But as Nicole was saying, I'm in Talca. That's 250 kilometers south of Santiago, in a really cool place called the Center for Integrative Ecology, and we do a number of things, including human microbiome research. There we go. So asthma is really important. It's a global economic burden with high direct and indirect medical costs. If you rank all the common chronic respiratory conditions, asthma is the top. It's the top worldwide, with 14% of children and young people being affected by the disorder. Asthma can and will kill you, right? So World Health Organization has recorded over the years around 495,000 deaths per year, mostly because of asthma attacks, but also because of some cardiac arrest and cardiac conditions indirect from having asthma. Okay, so I said that asthma is caused by different causes. There are a number of laws I recognize as contributing to the phenotype, but we also recognize a big role for the environment, right? And when we talk about the environment, we see epidemiological studies showing that, for example, in developing countries with large family sizes, a lot of contact with animals, livestock, pets, wildlife, you see a low burden of allergic disorders, right? This could be asthma, eczema, allergic rhinitis, and things like that. And on the other side, you can think of westernized countries with small family sizes, more urban, kind of more isolated from nature and wildlife, with high antibiotic use. And you see that the epidemiology there for asthma is the prevalence is increased, right? So this is not new. I mean, this figure comes from a paper published in 2001, but the so-called hygiene hypothesis has been around for decades. And we recognize that there is a role for the environment and of course microbes, right, in the development and sometimes the onset of asthma and other allergic disorders. There are some really cool pre-sequence everything studies, like the one here. This is a table from a paper published in 2007, and it shows the hazard ratios for for having, it's only the presence of bacteria. This was done by culture. The presence of bacteria in cultures from the airways of babies of 1 month of age, right? It's a prospective study, and then they saw whether or not they developed asthma or required hospitalization, whether they had some exacerbation of wheeze, persistent wheezing, and so on and so forth. And there is a lot of information here, but the only thing that I want you to recognize is that we have a list of usual suspects, right? And they have different hazard ratios for example, the first wheezy episode. You really don't want to have Moraxella catarrhalis in your airway if you're a baby, and so on and so forth, right? So we usually have four or five usual suspects that traditionally has been associated with asthma. And we started this in, let me see, maybe 2013 with Marcos and Robert at Children's National. And Robert had this really nice data set, metatranscriptomics of 18 individuals, right? Asthma patients and controls. And he said, can you find something here that is going to point out to a microbial effect, right? So we took the reads. This is expression data, right? So it's the microbial abundance is kind of confounded with presence of the microbe, right? It's we like to call it the active portion of the microbiome, right? And we saw this picture, right? Where on the right you have the asthma individuals, right? And in green, you can see Moraxella catarrhalis, right? And Marcos and I said, okay, we have something here, right? We went back to Bob, and he said, yeah, that's not really new, you know? It's not really important because we already know that Moraxella catarrhalis, among others, are exacerbated or higher in abundance in patients, right? So we said, okay, but we have a metatranscriptomics, right? So why don't we look at the host mRNA, right? And lucky for us, there was a gene expression signature for Moraxella catarrhalis that has been published, right? So we took that, and those are the genes that you can see on this heat map. And we asked whether there is a correlation for people with asthma and controls and the relative expression of these genes that are involved in Moraxella catarrhalis, right? And we can see clearly on the right side of the panel, these are host genes, so human genes. I forgot to mention that these are children and adolescents. And you can see so red is higher expression, right? Blue is lower expression. And you can see clearly, with some exceptions, maybe here, right, an increase in gene expression for the genes associated with Moraxella catarrhalis infection. So that was kind of cool because we were able to associate, on the one hand, active microbiome fraction, right? Really exacerbated, really abundant in Moraxella catarrhalis. And on the other hand, we saw a specific gene signature for Moraxella catarrhalis. So we like to think that Moraxella is actually mounting an infection, right? There's lots of ifs in there, so we can discuss that too. But overall, right, in general, how does the host respond? And again, this is a heat map. It's actually by recovering the reads that are human, not microbial, which in this case is really easy because these are nasopharyngeal swabs. So most of the reads that you get are actually human. It's really like a transcriptomic study with a portion of microbial, right? So red is upregulated and blue again is downregulated, and we see 79 genes that were expressed more than 8-fold in asthma patients in comparison to controls. Over a change in expression of 499 genes in total. So what are these genes doing? And lo and behold, most of them, 43, are regulators of pathways that in some other studies have been associated with asthma. They have to do with cellular and tisular proliferation, immune response, and inflammation. And some of them actually have experimental evidence in mice and some epidemiological evidence in humans. Okay, So we use the microbial reads to talk about the active microbiome portion, right? But that's expression data. So we can actually look at the functions encoded by those genes, by those microbial genes. And of course, we see differences. Here in blue, you see asthma functions associated with the microbiome in asthma patients, and the yellowish dots and bars correspond to the controls. And you have a number of things that actually make sense in the context of asthma, right? Very general things, but also things that have to do with asthma specifically. And we can take some of those traits if you want, like on the top, we selected this one. It has a nice correlation with the expression of IL-1A, an inflammation marker, right? So we have adhesion, that'll be bacterial adhesion associated linearly with the expression of IL-1A. And we can do that with also with taxonomic information. If we take proteobacteria in general, Moraxella catarrhalis is part of the proteobacteria phylum. We also can see a direct correlation with interleukin 1A. Okay, so that was our first foray into asthma back in 2013 to 2015. And then we started working with people from Boston Children's, again with Children's National, George Washington University, and my previous institution, Universidad Andrés Bello in Santiago, Chile. And we tried to look, again, using metatranscriptomics, whether in a prospective study, whether having bronchiolitis, we know bronchiolitis is a major cause of hospitalization in infants, and it's also a risk factor for developing asthma, right? So we selected infants that consulted the hospital for bronchiolitis of less than 1 year of age. We managed to compose, if you will, a cohort of 244, or we ended up really successfully amplifying and sequencing the metatranscriptomes of 244. And we asked whether there is some sort of different endotypes. It's recognized that bronchiolitis, allergic rhinitis, and asthma, it's multifactorial, right? And not all people that present asthma, for example, actually exhibit the same phenotype, right? So lo and behold, we using some clever clustering algorithms, we detected five clusters, 5 groups, different. You can see them here in this is a PCA version. It's a PCA version of a TSNE. It's an alternative to the traditional PCA, right? And you can clearly see the 5, we call them profiles, right? So yes, are there metatranscriptal profiles associated with bronchiolitis? Yes. Is there any relationship to asthma development? We'll see. So we follow these people from 1 year of age or less to 5 years of age, right? Before I tell you that, I just wanted to say that the profiles were really, I mean, how do we know they're real besides just the sequence similarity that is associated with clustering them, right? And we know that because because the profiles actually have a narrative with the patients, right? For example, profile B, you can see them here on the top left in orange. They have a lifetime history of antibiotic use, a history of eczema, parental asthma, and parental eczema, right? There is another one that is kind of interesting. In pink, you can see here on the top right, a high proportion of parental asthma and respiratory syncytial virus infection, right? We can see also that rhinovirus is a risk factor, and when we look at in green here, profile E, there is a very high proportion of rhinovirus C, but not rhinovirus A, right? So there is an association with clinical and virological factors for these profiles. And of course, there is some relationship to the usual suspects. So in these box plots, violin plots, we have the profiles here as a function of the presence of different microbes that we know are somehow and to different extents associated with asthma. And we can see the usual suspects, right? For example, profile E is highly associated with Moraxella catarrhalis, the one that we found in the previous study. Okay, so what profile, metatranscriptomic profile, that is, you don't want your children to have? So we estimated odds ratio. This is after a follow-up of almost 5 years, right? And we have the profiles here in this table. So the profiles are we like to mark them as with some of the features that define them, right? For example, profile A, it's characterized by the presence of RSV and some microbiome commensals, right? And we use that one as a reference. But let's jump to profile B and C, for example, that are highly associated with developing, I should say, a high odds ratio of developing asthma, right? And we see that, of course, there is RSV, but there is also rhinovirus A. And we see our usual suspects, Haemophilus influenza, Streptococcus pneumonia, especially those two associated with the high odds ratio. And we see RSV and Moraxella non-liquefacients and catarrhalis moderately associated with a high odds ratio of developing asthma. Okay, so do we observe similar patterns in microbiome composition in other populations? This is all done in the U.S. population, mainly Washington D.C. and Boston, the metro area in Boston. And so we look at this population in northern Portugal, in the city of Porto. We collected 347 nasal samples. This is a different type of cohort because in the US we were working with children and adolescents, infants even, but this is more like adults. It's a mix, but it's skewed towards adults, right? We still see the same usual suspects in high abundance. This is amplicon sequence data, though it's not metatranscriptomics. And we see different patterns of the diversity, some of which are not significant and some of which are significant. Here you can see controls. These are asthmatics. But one of the most important or interesting groups in our cohort here in Porto is the people with asthma and also with allergic rhinitis, because those things in adults at least tend to be highly correlated. So there's an increased likelihood of having allergic rhinitis if you have asthma and vice versa, right? We can see in this PCOA that we can tell apart the controls from almost everything else that is kind of a mixed in the study. But we were able to retrieve the usual suspects as in the previous studies. There's a mistake here. This should be the Chilean population. In this case, we collected over 140 paired nasal and oral samples. Usually and internationally, people use nasal swabs or nasopharyngeal swabs as a proxy for the bronchi or the lungs, right? It's not easy to get controls if you have to perform bronchial alveolar lavage on healthy people. You're never gonna get an approval for that. So the proxy oftentimes is nasal, and here we took
out of time. Sorry. So
okay, it's my last one. I just want to say that we retrieved the usual suspects.
I forgot to mention. Great.
And thank you for your time.
Thank you very much. Thank you very much.